and phenylactate, and it excretes in the urine. Guys, the odor of urine of these babies is like mousy odor. You know, the urine sample of that have a smell like a mouse. Mouse. Cat and mouse.
You don't know the smell of mouse.
Don't you?
How do you smell a mouse?
In our laboratory, I am working with mice. For example, for 1 month, I'm working for 100 mice with each other. I should at first induce cancer to them. After then they become cancerous. I will work with them, and they have a special smell. You can understand that after working with them. When I say something, at first it is so difficult. I was the person that I scared from each live creature, especially human. Okay, but excuse me. But because of my work, I had to work with mice. And after I think 4 or 5 months, I started working with them completely, actually restraining them, garbaging them, and doing some treatment on them.
You make maps. Monoclonal antibodies.
It is working for them too, for producing monoclonal antibodies. We should actually induce a seed in them. After that, in the peritoneal area, we can gather the monoclonal antibody from them. We work on that too. And you can see on those situations in animal health, we can be familiar with mousy odor. And the urine of these babies have mousy odor. Right. Phenylalanine, it's converted to tyrosine under the effect of enzyme under the name phenylalanine hydroxylase. I told you that hydroxylase need vitamin C. And another thing, there are a cofactor under the name dihydroxyurea that convert to tetrahydrobiopterin. I don't want you to memorize this, you will study, but I want to tell you, if the enzyme doesn't work, or these cofactors being interrupted and destroyed and being inefficient, they can't, phenylalanine can't convert to tyrosine. We have tyrosine deficiency an awake cycle. On the other hand, researchers actually show that melatonin have role in migraine disorder, but it's not completely the pathway. It's not completely known, but the melatonin have role in the sleep-awake cycle, and serotonin have role in happiness somehow. Have you ever heard about sertraline pills? Yeah? Guys, the sertraline pills actually inhibit the reabsorption of serotonin. Imagine. Guys, a central or sertraline is a pill for somehow sedative pill for being calm. Okay, antidepressant. Okay, imagine this is your neuron axon terminate that produce serotonin. These are for purpose serotonin. And here is another terminal axon for affecting serotonin here. This sertraline drug inhibits the reabsorption and absorption of this serotonin. The amount of that increase here and make the depressed person more happier because of the increasing serotonin level. It is related to guys about serotonin and melatonin. You should know that the precursor of them is tryptophan. Memorize that. Another amino acid, guys, it is not aromatic structure. It is basic. It is better to ignore about being aromatic. It is somehow aromatic, but not completely. And in physiology pH, it can have positive charge. It can have positive charge. And the ring is imidazole. It's not important. And you should know that the histidine is the precursor of histamine. Where do we have histamine? Mast cells. And in allergic responses, yeah, or in hypersensitivity situations. And another exception for amino acids, this is proline amino acids. Guys, you can see it doesn't have usual structure. It is somehow in a closed ring. You can see here. It is an amino acid, not amino acid. Amino acid. Guys, these proline in protein structure cause bending. What we are doing? For example, imagine this is tryptophan, lysine, aspartate. Here, proline, because of the special structure, cause bending in the protein. For example, here we have bending, and we can see here we have proline. Okay, you should know that they cause bending in three-dimensional structure of protein. I will tell you in tertiary structure. And again, the hydroxylation of them is so important, especially in collagen. Because let me say about physiochemical properties of amino acids. I know that you studied about titration. Am I right?
Yes.
With Dr. Sager.
Yes.
What do you remember? What do you remember about titration? Actually, you try to evaluate the buffering property. Am I right?
Yes.
What is buffering? Yeah, resist against changing in pH. Guys, at first, about proteins, I want to tell you about isomerization or optical activity, that it is related to asymmetric carbon or chiral carbon, and about buffering. At first, guys, imagine it is a mirror and you put your amino acid here. Guys, based on that your amino group be in the left of carbon or in the right of carbon, you have L amino acid and D amino acid. Can you guess which of them are predominant in our body? I told you L-alpha amino acid. L is in the body and D is in the nature. I told you we have 20 L-alpha amino acid in our body and we have another types of amino acid in the nature, in plants, in bacteria, in antibiotic structures and other places. This was about L-alpha amino acid. Guys, when you put a structure in front of a mirror, we call it an enantiomer isomerization. Do you study carbohydrate structure?
Yes.
And you know about enantiomer, yeah? This is enantiomer of each other that L-alpha amino acid we can see in our body. Guys, about buffering. It can be related to neutral amino acids like alanine, like cysteine, like glycine, and blah blah blah. And we have another acidic amino acids that have acidic and negative charge. And we have another basic amino acids that they are base and have positive charge. Guys, about neutral amino acid hydration. Look at this. You've studied that, am I right? Could you explain these to me? Look at that, think a little, and help me to explaining this. If there was volunteer, tell me. Is there volunteer? If there isn't, tell me that I start to explain. Do you need more time? Start. So we have anybody there?
We only know about the buffering region.
Yeah. The buffering zone is so important that we have it. Guys, about amino acid. I'm speaking about a single amino acid. When you are guys, imagine. This is a peptide chain that consists of different amino acids. Am I right? I will tell you in the continue. But here we have one free NH2 here and one free COOH here. Okay, in this situation, guys, just this COOH and NH2 can be ionized. The other NH2 and COOH are involved in peptide bonds. They aren't free. In amino acids chain, we have just these two parts. And if we have, for example, here aspartate, inside chain we have another carboxyl group. Am I right? Or if we have here arginine, we have another additional NH3 plus group. Okay? That can be ionized. Guys, I am now telling just about a single amino acid, not in a chain, that the carboxyl end and the other amino ends are free. Imagine you are an amino acid solution that you wanna add hydroxyl gradually. Okay? And you want to evaluate the pH. This is glycine. At first, guys, in low pH, the concentration of proton is high. Am I right? For example, in 0 pH, you have lots of proton in your environment. Am I right? pH is related to H plus concentration. If H plus concentration be high, your pH is lower. Am I right?
Yes.
Okay. Guys, in this situation, all of your molecule is fully protonated. You can see COOH, NH3 plus. Am I right? Here, your molecule have positive charge. You start to adding hydroxyl. Your molecule is a buffer. Amino acids have buffering role. It tries to neutralize the pH changes to resist. What did it do? It starts to releasing these protons gradually, and this is the area that it is releasing proton and becomes neutralized by this OH negative charge. And you can see here the buffering zone. The pH doesn't change dramatically. Okay? Here, pKa1 is related to ionization of these carboxyl groups. And it is the place that 50% of molecule is in this form and 50% of that is in this form. The pH suddenly increased where the all of COOH become deprotonated. Okay? The buffering zone resistance somehow disappeared. Your pH increased suddenly, and what is happening? Again, your amine group starts to releasing H positive or proton, and you can see another buffering zone again that is related to the NH2. Did you get that one? Yes. Am I right? And now you can see here we have another pK or constant dissociation that is related to NH2, and this was related to COOH. Guys, here we have a P isoentry. What you can see in this area, could you tell me? The charge of molecule is neutral. Yeah. The charge of molecule is 0 in this area. And guys, the P isoelectric is the average of these two pKs in glycine. Now, I want to make it a little complicated, more complicated. Here is the titration curve of an acidic amino acid that in the side chain have another COOH. Okay, to NH2. We have 3 buffering area, and the p isoelectric is equal to pK1 and pK2 that is related to those acidic parts. Did you get the point? It is true about basic amino acids too. Could you tell me the p isoelectric for a basic amino acid is where? Imagine this is related to COOH. This is related to NH3 plus of your chain of your amino acid, and another area is related to your side chain. And your p isoelectric is average of these two.
7. Around 7.
2 and 3.
Yeah. The steep part.
Yeah, around 2.
Here.
Yeah, exactly. If your amino acid was basic, the p isoelectric could be here, okay? And guys, the formula for neutral amino acid is pK for carboxyl group and pK for your amine group, average of that. For acidic amino acid, pK of COOH groups, you should average of that, and basic amino acid, the pK of amine groups. Okay? Was it clear? Crystal? Okay. Guys, about blood pH, I think my colleague told you, yeah? That the pH level of blood is so important to be a narrow range, and the decreasing of that is acidosis, and the increasing of that is alkalosis. And proteins, guys, have important buffering role in our body. Okay? Guys, let me check the attendance list. After that, say a little thing about proteins, and start after that, rest and prayer. Asra, Fatima.
She's absent.
What?
Because she has some problem at university, she will return her.
Ibrahim Abbas. I should read full name, or if he is asked.
Ibrahim Abbas.
Ibrahim Abbas and Delphi Muqtadi.
Ibrahim Abbas.
No, Ibrahim Abbas and Delphi Muqtadi. Ahsan. Irshad Muqaddas. You have to be students at all. Al-Baqra, Zainab, Mazhar, Azman.
It's maybe this for another
Yes,
person.
maybe it's for
us.
another class.
It's not in another class. It's our classmate, but they are some are not coming here.
This is your class.
Yes, that's right.
And Zain Nisa.
Me.
And Safar, Abdullah, Leva, Muhammad, Jawad.
Yes, he wasn't here.
One out of the class. Where here?
Yes,
Yes, he
yes,
did.
but now he's in.
And Farji, Abdullah, Bahassan. Amjad, Kamar, Fatih, Ali, Muhammad, Ali. Your name is so we will work on classification of proteins as our friend mentioned, and we will discuss a little bit about some disorders that are related to proteins. Guys, proteins have a structural role, like collagen, like elastin, like keratin, and some in our skin, in our head, in our nails, in all parts of our body we have proteins, structural proteins. On the other hand, in our immune systems, we have what thing our immune system produce? Immunoglobulins or antibodies. Okay? These antibodies and cytokines are made from proteins. On the other hand, they have role in coagulation. Do you know about coagulation? Yeah, fibrous protein that prevent bleeding. Okay? They have, again, they are proteins. On the other hand, the proteins can work as transporters. Do you know about one protein that carries lots of things? Hemoglobin carries oxygen. And not albumin. Have you ever heard about albumin? Albumin is a public transporter, general transporter. That, for example, lipids is carried by lipoproteins, but albumin can help the carrying free fatty acids, can carry some drugs, can carry bilirubin, and some hormones. On the other hand, enzymes can be protein, can have protein structures that have role in metabolism. On the other hand, as our friend mentioned, the hormones are made of proteins that have role in regulation of our body. On the other hand, guys, have you ever heard about actin and myosin and tropomyosin and troponin? Yes. They are proteins that have role in our movements. Okay? They have a, guys, just our physical movements. No. Which type of movements do we have? Yes. Yeah. Some peristalsis and in smaller scale, in cell division. Here we have one cell, am I right? Start with mitosis and cytokinase, am I right? Some microtubules is start to produce here to have cell division. These are again some proteins related to movement in molecular scales. Okay? And also they have storage role as we have in ferritin. Do you know what is ferritin? Ferritin is the storage form of iron. This is the storage form of iron. You can see that when a medical doctor, a physician wants to evaluate your anemic situation, at first they will evaluate CBC, stem blood count, that in this situation they evaluate hemoglobin, RBC, YBC, and something like this. Another important issue is your ferritin. It is the storage form of iron. If your ferritin storage be low, it is an alarm situation that you will going through being anemic. Okay, this is about them. So proteins can have storage role as ferritin, and they have role in some diseases like Alzheimer's, like Parkinson's, like some metabolic abnormalities that I mentioned. Good. About hierarchical structure, you mentioned sometimes. At first, we have primary structures, secondary, tertiary, and quaternary. Okay? At first, about primary structures, guys, just we chain, but in quaternary structure we have more than one chain. Okay, I will explain you. And here we have some structure that I tell you, and in the tertiary structure we have three-dimensional structure. Now, let's review. Guys, this is primary structure that amino acids units are actually attaching to each other by peptide bonds. Okay? After that, these amino acids chain start to have some folding that it occurs based on hydrogen bond. Okay? We have secondary structure here. And then we have some more folding that we have tertiary structure like myoglobin. And if your protein have more than one monomer, more than one subunit, we call it polymer and quaternary structure. It is what one? It is the structure of which molecule? Do you know? Hemoglobin. Okay, this is myoglobin that it consists of just one of these subunits, and this is hemoglobin. Guys, about primary structure, as I've told you, we have just a mild bond, that they are covalent bond. Guys, the structure in this situation is linear, zigzag, monomer, and one chain. In this situation, we have N-terminal and C-terminal. C-terminal is related to carboxyl and N-terminal is related to amine. Another thing, guys, when we want to number one protein chain, we start from N-terminal to C-terminal. I will show you. On the other hand, the product of translation is primary structure. Okay? At first, primary structure is produced, and after that, start to produce secondary and tertiary structure. And this is the backbone of protein structure. Guys, in this structure, R have no role. R in tertiary structure have role. In primary and secondary structure, R have no role. Guys, what do you think? The order of amino acids in primary structure make the folding of other structures or not? No. For example, if your amino acid order here is the folding of that can be different by the order of amino acids are important for folding or not? Yes. It is important and critical. Okay, I will tell you and show you. Guys, the primary structure is determinant of other structures. The primary structure make that how other structures form. Guys, here you can see the peptide bond that occurs between carboxyl group and another amine group. Here you can see that we have production of a water molecule, and we have here peptide bond. Here, this is a dipeptide. You can see this is N-terminal and this is C-terminal. Guys, what is the importance of this N-terminal and C-terminal? Why I am saying that we named our polypeptide chain from N-terminal to C-terminal? Why? What is the importance of that? In transcription, and another easier thing.
And to name the protein because N is the more valuable or strong, N is the most important because that's where you start, like 1, 2, 3, 4. That's what
Do you know, for example, I want to tell you about the order of amino acids in insulin structure. It is the same in all of us, am I right? If I want to tell you that the insulin is made up of these amino acids, I start to name it from N-terminal to C-terminal. Okay, it is somehow an international consensus that it is better, for example, my name is Faton. It is somehow a consensus that I am Faton. But for proteins, there is a consensus that you should name it. For example, say insulin is made up of adenine, glutamate, valine, isoleucine till the C-terminal. On the other hand, guys, be aware, this is NH2, COOH, chiral carbon, and side chain. The side chains cannot be beside each other. Okay? The form of that is somehow in transform. Because of what? What is the reason of this arrangement?
Steric hindrance. That's what they call it. They repel each other. Right? The steric hindrance. In chemistry called steric hindrance. If the groups are on same side, then the polarity arises and it disturbs the structure.
Yeah, somehow it's become produce some limitation for each other and some space limitations. Okay, this was related to peptide bonds. Guys, about secondary structure, there are lots of hydrogen bonds. Do you know about hydrogen bonds?
Yes.
Are they weak bonds or powerful bonds?
We are weak, but there's a lot of bonds which makes it strong.
Yeah, a lot of them start to be strong, and the stability and flexibility of proteins depends on hydrogen bonds. And secondary structure is based on them. Again, I have no major role. Okay, could you tell me that the H bond is between which groups?
Oxygen and hydrogen.
From where?
More electronegative groups. If there is an electronegative.
Here you have a protein amino acid structure, okay? And another amino acid here. Could ionic bond between an acidic amino acid